This is deoxyribonucleic acid, more commonly known as DNA, a molecule so important that it is in every single cell in our bodies. It carries all of our genetic information and is what makes us, us. But if DNA is the same in every cell, then why is a skin cell different from a bone cell, which is different from a liver cell? The DNA is the same in each cell, so the cells should all be the same, right? This is where epigenetics come in. Epigenetics are heritable changes in gene activity that do not change the sequencing of the DNA itself. Proteins, chemical processes, and DNA modifications which affect gene activity are all related to this process. It is what causes an active gene versus an inactive gene. One easy way to think about epigenetics is writing a book. The written text is the DNA, while the editor, deciding what stays in the book, are the epigenetics. Epigenetics uses several different mechanisms to affect gene activity. The first, DNA methylation, is a biological process where specific naturally occurring chemicals called methyl groups are attached to the outside of the DNA molecule. This attachment can then influence which genes are expressed. Another process, histone modifications, are when proteins called histones, which DNA coils around to store itself, are used to turn off or silence genes. Based on the way the DNA has been coiled, whatever gene is stored can become inactive. And finally, non-coding RNA. RNA is the nucleic acid which is mainly used as a messenger to make proteins using the DNA's instruction. Non-coding RNA is the RNA which are not translated into proteins. While not much is known about these processes, many scientists believe that issues in them can cause genetic disorders, metabolism issues, or even cancer. Some scientists even think that epigenetics could be used to reverse aging. While a lot is still to be discovered, it is clear that the ability to switch on and off genes play a very important role in the body.